than the actual tissue if you have some squeamishness about that. And the surface of the testes has a connective tissue capsule known as the white coat or tunica albuginea. And that connective tissue capsule sends septi or bands into the testes itself, dividing it into lobules. So this capsule is known as the tunica albuginea. Tunica means coat, and albuginea, like a albino, is white. And then these would be the septi, and a lobule. So each individual lobule is going to have about 16 to 20 seminiferous tubules. Those are hollow channels lined with a simple columnar epithelium in this pattern. So we have a seminiferous tubule joining to form a straight tubule, and another seminiferous tubule with another one coming together to form a straight one, in that fashion, 16 to 20 of them. Right? Now I'm going to take this segment here and draw it a bit larger. So when I diagram and name those structures and pathways, it'll make more sense. So if you don't mind, I'm going to erase this image and just take a segment of that and draw it more large. So each seminiferous tubule is highly coiled, packing more surface area in there, which means that when we cut the testis and look at it, we may be cutting the same tubule multiple times if we see multiple rings. Then the two tubules come together to form a single pathway. So that one of those would be a seminiferous tubule, these structures here. Right? Here we have two seminiferous tubules joined together to form a single pathway. That single pathway is identified as a straight tubule. In Latin, tubuli recti. You happen to see that word, it's the same thing, but for the lab quizzes and exam, straight tubule is fine. And then all the straight tubules join together to form a network of interlacing branched structures known as the reedy testes. The reedy testes and straight tubules are not the same. Tubuli recti. They both have a T and an R in their name, but they're not the same structure. And these are considered components of the testes, the gonads. The reedy testes are then connected to the epididymis by the channels we looked at, the efferent ductules, when we looked at the video of the developing testes. And so we would have multiple channels that then join another highly coiled structure, which is the epididymis. These channels are the efferent ductules, refer to them just as efferent ducts. And then finally, this structure is the epididymis. So over on the diagram, we would have the seminiferous tubules, the straight tubule, these short segments here, the reedy testes, efferent ductules, and epididymis. So efferent ductules and epididymis develop from the mesonephric duct. The other structures are developing from the gonadal ridge and are components of the testis. And it's within the seminiferous tubules that the sperm undergo mitosis and meiosis and formation of numerous millions of spermatozoa. From there, they enter the lumen of the seminiferous tubule and are propelled through all of these structures to the tail of the epididymis. They don't swim yet, but they are propelled through by these have epithelial cells that have contractile components, as well as the addition of fluid, and that kind of just sweeps it along. Yes?
So this efferent ductule is just the bottom part of the epididymis?
The efferent ductule are these segments right here. So they're separate components. The epididymis is only one. So I don't know what you mean by the bottom part. The tail of the epididymis is down here. Right here.
Because it's it looks like it's just one and it's just getting bigger from here.
The head is actually up here.
Okay.
And then all of these are the efferent ducts.
Yeah, they said the head has got
The whole
a bracket right there.
It's seeing that part, but it's really just a single duct structure. All right, now let's look at what this looks like as developing sperm. And so if we look at, this is a high magnification, not actually high power, but high magnification of about 2/3 of one seminiferous tubule. Okay? So you can see the individually developing sperm cells. I have a slide, a lower power view here. So this would be the testes over here. So these all might be the same seminiferous tubule, but because it coils so many times, it's cut through multiple times. There's very little interstitial tissue between them when we compare it to the epididymis, as we will do later. And what is found there is going to be the site of the cells that synthesize and secrete testosterone, the Leydig cells, or the interstitial cells of Leydig. So we're going to take one of those tubules and look at it at a higher powered view and follow, not meiosis, we'll do that Thursday or Friday, but at least the cellular changes and development as we move through the seminiferous tubule. Now, I said the seminiferous tubule is formed of epithelial cells. So we're going to come back to this and kind of go back and forth between the two images. So let me explain the background for that. May I erase this illustration? Okay. I want to give you the background. We tend to forget the presence of the epithelial cell that forms the seminiferous tubule because all we can see are the developing sperm cells. So if this is a transverse section of one seminiferous tubule, and we leave out all the developing sperm cells, what we would see is a simple columnar epithelial cell, its nucleus, and these are the cells that form the epithelial lining of the seminiferous tubule. They are identified as the old name, Sertoli cell. Apparently, we're not supposed to call things by the name of the people that describe them anymore. So the newer name is sustentacular cell. Sometimes nurse cells, their role is to provide the proper environment, maintain the high concentration of testosterone, which influences the development of the sperm. So all of these, except for this one and this one, the rest of these nuclei are the developing sperm. They are not in the cytoplasm of the sustentacular cell, but are separated from that by plasma membrane. To explain, remember back in Bio 430 when you learned about Schwann cells and how they myelinated axons by wrapping around them tightly? And even unmyelinated axons were still associated with Schwann cells. So if this was an axon of a nerve that was not, several nerves that were not myelinated, the Schwann cell membrane was still intimately associated with it. In this fashion, even though it didn't wrap multiple times around it. So when you look at an electron micrograph of a Schwann cell, you would see these axons that appeared like they were inside a Schwann cell, but they really just had the plasma membrane wrapped around it. Okay? And that essentially is similar to what happens with the developing sperm cells and the sustentacular or Sertoli cell. So let me illustrate. Now I'm going to be making I'm going to erase and draw and erase and draw, so you might want to wait or follow along, maybe take a picture of it when I'm done, rather than trying to copy every line that I draw. So I'm going to take one of these sustentacular cells and enlarge it. Kind of like you see it, but I'm going to flip it around because I prefer my lumen to be at the top rather than at the bottom. So here would be the lumen of the sustentacular cell. Here would be another sustentacular cell over here. A basement membrane, because it's epithelial. And along the basal surface of the sustentacular cell, we find the least modified, the least changed of the developing sperm cells. So when the boy is born, his testes are full of spermatogonia. And again, we're going to talk about meiosis on Tuesday and or Friday. So here would be a spermatogonia. And I'll write the terms up over here on the side so it doesn't compromise and make the diagram too messy. So spermatogonia is the source, if you will, of all the developing sperm that are going to be produced. They are diploid cells. In other words, they are going to have all 23 sets, 46 chromosomes, just like every other cell in your body. They undergo mitosis to make more spermatogonia. One will develop and further differentiate and undergo mitosis. The other one will remain behind to undergo mitosis again, so that we always have a pool. Okay? So I'm going to leave that one there and describe, I'll leave that one there and describe this one. So this one's going to divide and undergo mitosis and make another spermatogonium, which will also be surrounded by the plasma membrane. And then one or the other of them, randomly, is going to further change to become a mature spermatozoa. It doesn't yet divide, but it migrates up into the cytoplasm and becomes a larger cell. The chromosomes duplicate as if it's going to divide, because it will in a short time, several days. And those chromosomes become rod-like structures visible within the nucleus. It maintains the plasma membrane associated with it. And so this cell, it hasn't divided, all right? It's just doubled its chromatic material. This cell is now identified as a primary spermatocyte. Let me show you what it looks like in an actual histology. So here would be spermatogonia. They're always along the outside edge of the seminiferous tubule. These are the primary spermatocytes, sometimes called bouquet cells because of that blossoming of the chromosomes. So they've doubled and then condensed, so they become visible as these rod-like structures. So all of these are primary spermatocytes. One spermatogonia becomes one primary spermatocyte. And then it makes its first meiotic division. So one primary spermatocyte is going to divide. These actually remain attached to each other. Remember, this cell is becoming this cell, okay? So that cell is not going to be there anymore. Another cell may move up and take its place, but this cell is becoming this cell. And we call that, so one primary spermatocyte is going to form two secondary spermatocytes. Again, we'll talk about the chromosomal changes that occur later. I can't show you a secondary spermatocyte in this histology slide because they only stay a secondary spermatocyte for a very short period of time and then they immediately divide again. So the probabilities of capturing one are very slim. So you might see it in the diagram, like the previous one, labeled such, but in a histology slide like this, you won't see it labeled. All right? So in other words, secondary spermatocyte will never be a correct answer for a histology question.
Good to know.
Now, the secondary spermatocyte immediately divides again. So each one is going to form two cells. Again, cytoplasmic connection. And these cells have very dense, small nuclei because they're no longer synthesizing protein and the nucleus is condensing, ready for transport to the female reproductive tract. But still surrounded by the plasma membrane. So they're inside, kind of, the sustenance cell, but not in the cytoplasm, okay? It's kind of like you took some bread dough, rising bread dough, and smushed it over something. You can see it in there, but it's not actually mixed up with the dough. It's kind of just overlap. So each secondary spermatocyte is going to form two cells identified as early spermatid. No more cell division occurs. No more mitosis. Instead, the early spermatids, which are these cells, let me turn the lights on. These cells.
Look like perfect circles.
Yeah.
Solid.
They're small, dark, and round, and condensed. They start to lose cytoplasm and take on the appearance of a spermatozoa. See these angled heads? They're still embedded, associated with the sustentacular cell. They're not free in the lumen. So as long as we see them in this relationship, like an ostrich with its head in the sand, they're late spermatids. Okay? They have the same genetic component as the early, but this, a different physical appearance. Now they have the tails, they have the angled heads, but they're going to be still in this association with the plasma membrane. And late spermatids. They're going to look the same, but when they're free in the lumen, we now can call them spermatozoa. Spermatozoa and its single. Okay? So we'll come back Thursday and Friday, and we'll go through the same steps with the developing oocyte and talk about the actual genetic changes that occur. Your quiz on Thursday will involve a slide of the testis, but I'm not going to yet ask you to identify those different cellular stages. I just want you to be able to identify it as a testis. Okay? Certainly later on, next week, you'll need to know, is this a primary spermatocyte or a secondary is not going to be an answer, early spermatid or spermatogonian. So just recognize it as a testis for now. Yes?